which are scientists working on product development, production, and process development to develop this manufacturing process that we have for manufacturing stem cell lines.
What we've done here at CDI is take that process and really take it out of the laboratory and put it into an industrial environment that then can be shipped frozen all over the world to be studied as a true human surrogate for biology.
In science, whether there's a eureka moment you can point to and what your feelings are comes up pretty frequently. And in the case of human ES cells, I physically did that work myself. When you take a cell from your skin, for example, and put it into tissue culture, it'll divide well, and scientists will figure out how to do that. But it divides a set number of times that undergo something called senescence. But with these embryonic stem cells, you can divide forever, so you can make as many as you want. So that's the first property. The second property is that they'll form anything in your whole body.
Those had political and ethical concerns around them based on their derivation. Many of the researchers, including Dr. Thompson, started to look for alternatives to represent individual biology. We were able to take a blood sample, have it shipped to CDI, and we were able to reprogram his cells back into induced pluripotent stem cells, or iPS cells. We then expanded those cells into a large enough quantity such that we could put them into what we call the differentiation process. We then were able to make his heart cells in relatively large quantities and at a purity of above 90% such that we could really study cardiac behavior based on his individual biology. We then tested all of the electrophysiological properties of his cells to make sure that, in a sense, his cells beat appropriately, that when we put drugs on his cells that caused them to speed up, that his cells did indeed speed up, and when we took that drug away, his cells came back down to a basal normal level. We then followed that up with looking at a variety of different drugs to understand if there were other biological properties that were found in his cells that may predispose him to toxicity for certain types of drugs, but not necessarily just on the beating capacity, but also as it relates to drugs that, for instance, like cancer therapies that have been known to show cardiotoxicity, that we were able to recapitulate that in the dish as well. So in a sense, we now have a biological fingerprint of David's cells such that we could, in essence, if he was going to go on a new drug that could have effect on his heart, that we could test that prior to him taking the drug so that we can really study why individuals respond in the way that they do.